Welcome back everyone. Today we learn about gene transfer. Genes may be transferred between cells of the same species through asexual reproduction, or sexual reproduction between same species. But genes can also be transferred between cells of different species, giving rise to transgenic plants or animals. This is possible due to the role of bacteria, vectors such as viruses, and with advancement in technology such that genetic engineering is possible. We will look into how genetic engineering in the medical biotechnology field has benefited us. We will be looking at the production of human insulin using the E. coli bacteria. The general process of how this occurs can be applied to other scenarios as well in genetic engineering using bacteria. The first step is to isolate the gene of interest. The gene is isolated with the help of restriction enzymes that cut the gene out from the chromosome. The next step is to use the same restriction enzyme and cut the plasmid from the bacteria. Plasmids are small extrachromosomal DNA molecule within a bacteria cell that is not part of the bacterial chromosomal DNA. Once the gene of interest and plasmid is cut with the restriction enzyme, the gene of interest, which is the human insulin gene, is inserted into the plasmid. This is done with the aid of an enzyme, DNA ligase, which helps to combine both of them together. The resulting plasmid formed is now called a recombinant plasmid. Now, this recombinant plasmid has to be placed back into the bacteria, and this is done by subjecting the bacteria to heat, electric shock, or chemicals. Once the bacteria takes up the recombinant plasmid, it is cultured in a fermenter under suitable conditions. This is to mass produce the human insulin. Once done, the human insulin is isolated from the fermenter through purification process and insulin is thus manufactured. While genetic engineering seems like a good thing, there are social and ethical implications of it. First is the increased economic inequality, as only those who are wealthy enough can afford such treatment technology. Second is the contradictions in religion and beliefs. Should animals such as pigs be used in genetic engineering, it would be controversial for vegans and Muslims, which ends up restricting these people access to such technology. There could be unknown harm or allergies that arise due to such gene transfer. There can also be damage to the environment. And lastly, there could be unforeseen health and environmental hazards as a consequence of genetic engineering. That's all for today. Thank you for the effort to learn with me. Stay tuned for more interesting lessons. See you next time. Bye-bye.